Hello. Is Matt coming to this meeting? Does anyone know? Because I swear I saw him say that he wouldn't make it, but now I can't find the comment where he said that, and he's supposed to be chairing. So,
Yeah, I just found it. He's not coming.
okay. And he asked
Indeed.
us to swap, right?
I don't know if I don't see that coming. I just see, sorry, I won't be able to make today's meeting. If there's anything else needed, please drop a mention in the comments.
Which is that on today's meeting? You know what I mean? Like
I just see it in my email.
Yeah, see, I'm confused. I feel like I saw the email and like didn't open it on GitHub, and now I can't find it on GitHub. I am confused.
Oh no, that's not Matt, that's
Fantasy?
Fantasy. Yeah.
Yeah, okay, that's Hung. So I mean, he's not meant to be chairing anyway. I guess if no one's shown up by 5 past, then we will assume that I did not dream up that comment. I'm just so confused. Where was it? Okay, yeah, he's put himself as an apology on the agenda. So I think we can definitely assume he's not coming. I'm happy to chair, but then there's nobody else from UCL here to take minutes, is there?
I can take minutes, but I'm not from UCL.
Hi, Clementine. Sorry, I joined a few minutes late. Is Matt not coming today?
No, apparently not. We were just saying he definitely said it somewhere. Can't find the message. But I think he said something about there's like things changing at UCL, or I don't know. He has a lot.
Okay. I see.
Are you able to take minutes, Dimitri, or
Would someone be able to back me up because I'm commuting today and I'm at the airport?
okay.
Yeah,
We can swap next
back you
time.
up.
Next time I will do them.
Yeah, that's
Minutes.
right. So Junior, if you're happy to take minutes,
Yeah.
we can sort it out.
Yes.
Yeah. Let's get started then. Okay, technically, it's you and me who are meant to start off anyway, Junior. So with new results, I have a very short presentation. It's possibly not even worth making it a presentation, but oh well. Let me just
Let's first introduce Tim.
Oh
Tim
yes,
is the
some introductions because we have
Tim is the new postdoc in our group.
Yes. Hi, everyone. So my name is Tim Balse, and I started last week in the group of Wendy van der Sander, and I'm the new postdoc going to work on the MycetOS project as well.
Junior, do you want to introduce yourself again as well? Because again, we have some new people.
Hi, yes. My name is Dunya. I'm the PhD student who's hopefully making a model that can replace the larva work and test some new compounds that the chemists make here.
And Vijay, I feel like I saw a message from Matt as well, but do you want to introduce yourself?
Good morning. Sorry for interrupting. Good morning to everyone. I am Vijay Masan from India, and I am the new joining in this group. I'm working as a professor in a college. I'm really sorry with the audio with me.
That's all right.
Some problem is with my computer. Okay.
Yeah.
And right now I'm working in the field of molecular modeling, docking, QSAR, and others. And one more thing, I'm trained organic chemist with specialization in synthetic chemistry and retrosynthetic analysis. That's much.
Okay, great. Molecular modeling, especially, is good to have. Okay. I guess I will go back to showing you guys my beautiful little PowerPoint. Give me 1 second to share my screen. Second. Of course, my computer is deciding to not work. Okay. See, you guys are just seeing the presenter mode, aren't you? Not the proper one.
I think it's working now.
Oh, it is working? Okay, cool. Alrighty. So, quick background. I've been testing a couple of compounds, or a collection of compounds, some of which we made this year and some of which we made previously but hadn't tested yet. They're from series 2 aminothiazoles, and there were a couple of different things that we were looking at. So far I've done like the initial screening, which cut out quite a few of them. And then I am just finishing up the MICs. So these are not final numbers, and I haven't updated them on our big SAR thing because they're not finalized yet. But they're still, enough to go by in terms of looking at trends. So these were the ones that actually did anything at a high concentration. Then we looked at the MICs. I realize out of context, this isn't very helpful. So I tried to contextualize them a little bit here. So one of the ones that we were, that I've just tested was this one here with a fluorine in a slightly different position. And I mean, basically, we just seem to be seeing that the position of the substituent is not super important on that right-hand side. They, the activity is relatively similar wherever it is. The same thing with the methoxy. So this was one that we've tested recently. This year, we decided to make a bunch of these dichloro compounds because those chlorine ones had been so active. That didn't work out too well. So the only one of them has had actually good activity similar to our lead. So that's, annoying, but good to know. And then this, I think, is a little bit more interesting. Pretty much every single variation we have made on the left-hand side of our molecule, part that comes from the bromoketone, has not worked at all. Like nothing has worked. But we have finally, we realized that we needed this pyridine ring on the left-hand side. And so we've started making a couple of different analogs which have the pyridine ring and then also various substituents. And we seem to be seeing that once you've got that N back there, they do seem to work. We haven't made many of these, so it's kind of hard to say too much about it. But it's nice to finally have something work on that side. Oh yeah, the only other thing I had on here was the toxicity, which I've been doing a little bit out of order. But mostly they seem to not be toxic, which is nice. Hopefully I will manage to get some efficacy data before the larvae season ends, but see. I'm also, they're not on this slideshow, but I've got compounds from the University of York. I don't think there's anyone. Yeah, no, Peter's not here today. So I've got some series 3 compounds from the University of York and a couple of 2-aminothiazoles from them as well that I'll be testing. And I've also got some compounds sent from UCL, but they were made by Whitgift School, which I'll be testing in the next couple of weeks as well. And that's all from me. I feel like there's nobody here who works on 2-amino azoles, is there? Yeah. Okay. I guess we will simply move on. So more housekeeping stuff. With our master list, we had a whole bunch of tabs that it looks like people have made them for various projects at various times. And basically, if nobody has said, please keep this tab, it's important, I still need it, we are going to be deleting those extra tabs. So if you know you have something in there that you would like to keep, please tell us now. And in the future, if you are making a new tab, like if you're, copying data across because you're using it for a paper or something like that, just put in the first cell of the page, put an explanation of, who you are doing this, why you're doing it, and when you need it until, so that we can avoid getting too many of those tabs again. Any questions about that? And nobody has any super urgent data on there, I'm assuming. Cool. Again, so we talked last meeting about changing our, I guess, kind of how things are set up in terms of if people come to GitHub to look at the MycetOS project. So we had these two repositories that were just kind of sitting there, not really being used. So those are the general start here and the what other molecules repositories. So I have tried to kind of reshape them a little bit to make them a bit more accessible for people who don't know GitHub and don't know this project. So if you have a look at the wiki, I'm just going to put it in the chat rather than share my screen because I think that's a bit simpler. I basically just tried to kind of collate everything that is, unchanging, kind of like not a discussion, into the wiki. So for instance, we have a page that has papers that have been published relevant to MycetOS, and it would be good if we could also put theses on there. I know that some people think we don't have that many theses that are related to MycetOS published yet, but I think some people have them somewhere. I think Hung has his honors thesis somewhere. I'm not sure where it is. But Dimitri, for instance, if you would be happy to put your thesis on there when you have time, that would be good.
Yeah, sure.
Okay. Again, yeah, if you guys have a look, click through that. If you have any suggestions on how to change it, if you think there's anything that's not clear, just, leave a comment in this chat, like if you're looking at it later, and we can talk about it. But I think this one's fairly obvious. The one that's a little bit more confusing, firstly, I asked Matt to make this public project, but I'm not sure if it is. To me, it's saying it's not, but somebody else could have a look and open it and see if it tells you, like see if you can access it. That maybe will tell us whether it's public or not. Because this
gives an error for
private then. I think because Matt's the only one who can change the public-private setting, because some of us have like editing rights, but not all of the editing rights. Basically what I was trying to do is use the GitHub project management tab to organize the potential projects. So instead of just having like the list of issues that's just like, here's 10 different things that people have suggested, randomly, I was trying to organize them into kind of themes. So I will share my screen for this if you guys can't see it. So here, for instance, each of these are issues. So I've just taken the existing issues and put them into some organization. But there are some things where people, where we've just said, oh, it would be good if we could get this molecule. Does anyone know where we could get it? Right? So those are things we'd like to screen, we don't have access to. And I separated out things that are like, we would definitely have to make this synthetically. So synthetic chemistry, modeling, things that are looking at literature, other. Again, if you disagree, once this is public, if you disagree with where these are or what these headings are, let us know. But my approach was that if somebody is, looking at this project going, oh, is there any synthetic chemistry that would, be helpful for this project? They can kind of find it a bit more easily than having to open every single issue and go, oh, this is actually about, protein modeling, whatever. Okay. Again, if nobody has questions about that, we'll move on. Does anyone have any questions? So Laurie isn't here. I believe she commented saying that she would send us some compounds. Can anyone remind me if that's right or if I also dreamed that? I think maybe people have left some comments on the old meeting. That would make sense. Okay, no. Does anyone remember Laurie commenting on those compounds since our last meeting? I guess we'll leave that one for now.
Looking at the thread for the old meeting, I don't see any comments from Laurie.
Again, I'm a little confused. I swear I saw, because I get the notifications on my phone. I didn't open it entirely, but I swear I saw somebody saying, oh yeah, I'll send those compounds that you've asked for. And I can't think of who else it would be except for Laurie. So yeah, that's confusing. That's right. I'll see if I can find that comment later, but otherwise we'll move on. So phenaramols, I know Matt said that he hasn't finished everything he said he would do, but he's still working on it. Dimitri, do you have anything else you can tell us about where the paper is at the moment?
It's just an update on one of the threads that one of the objectives on GitHub page I did yesterday. So the desired compounds, I think Clementine, you were the one who set up this page. I just wanted to let you know that I updated it with a scheme and Smiles code. Is that what was missing there?
Yes, that's exactly right.
Yeah, so that's done now. It's the very first objective on the page.
Okay, thank you for that.
Other than that, as far as I know, Matt has the final version of the manuscript, so as long as he's okay with that, then we can submit it.
Okay, cool. And we discussed what the paper, what journal you'd be submitting it to last time, right? We don't seem to have written it down here, but do you remember?
Matt hasn't told me because I was submitting my thesis, and I missed a few meetings.
Yeah, I mean,
a few Mycetoma meetings previously, so I'm not sure yet because I haven't seen him for a while.
I think it was one of the ACSs, but I can't remember which one. That's all right.
But infectious disease.
Sorry?
ACS infectious disease, I thought was
that
possibly.
That definitely sounds.
There were at least two options as far as I remember, I think he was considering.
Okay. Well, I guess, I mean, we can move on to series 2. don't have a lot else to say, seeing as I've already...
Clementine, sorry, just a quick question on Series 1 phenomenals. So the molecules that you specified in that thread, those two, are they on high priority list at the moment?
I think you're probably better placed to answer that than me. My understanding was that they're not high priority, but it would be nice.
Okay, I'll ask Matt then, probably, just to save time.
I did see that Hung has left some comments about other compounds. So there was a page that was out of date about desirable compounds. So I can, update that and merge those two pages. But he's basically said that there were some compounds that previously you guys had said it would be good to test them, but some of them are no longer interesting because you've realized that the amine and amide motifs of the piperazole positions are inactive. So I think I should be able to make those changes.
Because Wendy previously commented on Series 1 phenomenals, and I think she made a point that they would not be testing them anymore. So
Yes.
That's why. And then those two compounds are appearing on the GitHub issue. That's why I asked, essentially. Okay.
So to go back to 2-aminothiazoles, as I was saying, I've already updated on what we have on the new things we've made recently. Erica has been doing some work on mechanism of action with her, I think they're master's students. And I know she's left a comment about that. She's got a document to explain it. I am going to leave that for now because I think it will be better explained by Erica. But she does say that they tested 3 different enzymes, and 2 of them are, worth exploring further. Again, I don't think anyone's got anything to add there, but if you do, jump in. Okay. Otherwise, again, Series 3, unfortunately, Peter from the Uni of York is not here, but he sent us a whole batch to test. Again, unfortunately, our preliminary results are that they are not looking good at all. I think, Anthony, you were saying that was a bit surprising, actually. Did you want to tell us more about that?
Because structurally they're very different than there were 2 that were active, you said, and I had posted those. And those were very different structurally than sort of the curaxin type compounds. So the curaxin type compounds have that tricyclic head with an isopropyl ethylamine tail hanging off of it. So so those don't have that. That one of them just has an acetyl group hanging off the end. I posted structures for the compounds that the 4 that you found. 2 of them were aminothiazoles, and then there was 2 that were thiazine derivatives, but one of them, interestingly though, it's kind of interesting that the MycetOS 752 has that sort of pyridine with a sulfur next to it, kind of almost like the aminothiazoles, which is quite interesting.
Yeah, those results that I have are still quite, mostly we are confident that we're confident about the ones that don't work. I don't know what the MICs will end up being for these 2. But yeah, that is interesting. I guess we'll leave it until we know a bit more about how active they are. But I believe Peter's going to be making more this year. So
yeah, the ones that are going to be more like the curaxin compounds that Laurie's group discovered.
Yeah.
Worked on. Because we know that the ones that Lori sent, some of them were potent.
Yes, that's the I've forgotten their numbers. It's like the 728, no, 628, that set, yeah.
So I think the next step is to go keep that tail portion and just change the head from the carbazole to other tricyclics to see what we can get.
Yeah. Yeah, it would be good if Peter could tell us what he's making next, but I gather they still haven't quite decided on it. In any case.
But I did just sort of in that I do have a desired compounds I worked on. So we're kind of, trying to keep the head of the trying to change the head, keep the tail of the Giraxins. Some analogs too, because I remember Lori said that the acetyl groups on the carbazole were giving her problems in terms of DMPK and when they were doing testing for trypanosomiasis. So it'll be so hopefully we can work out some of the bugs and see how it goes.
Yeah. And I do also see, less interestingly on our agenda that apparently the toxicity data, which was done earlier this year for those compounds sent by Laurie, apparently that's not been added to the wiki. So can anybody do that? Junior, would you mind doing that if I can show you where to put it? And then we can just...
If you show me, then I can do it probably today or tomorrow.
Agenda so that it's not there for 3 more months. Okay. Yeah, so benzimidazoles. Andrea, do you want to talk to us?
Sure. I've only been involved in the benzoxazoles, but I think they've been put under the do we call it 5B or something?
Yeah, we haven't changed the name.
Yeah. Oh,
Yeah.
okay. Yeah. So I've only ever, well, my students have only ever contributed the benzoxazole series. So I think we're starting to draft a manuscript on that. I think Wendy and Ma both aren't at the meeting. I've got a few questions, but I'll just put them in the started off a manuscript thread in the hub. So I'll just post questions mostly to Wendy and Ma in there. I'm happy to start writing up all the data of all the synthesis and NMR and stuff like that, but I just need to know what journal and stuff we're aiming for at least, or what family of journal, so I can get the formatting right and stuff like that. Yeah, I think that's really the only update there. We haven't, we sent compounds in April, May. I think that was the last batch of benzoxazoles that were tested by Wendy's lab. Yeah, I don't know if Charlotte at Imperial has made any this year. I think her students usually synthesize December, January, February.
Okay.
So I'm not sure if...
Sorry, are you planning to make any more this year, or are you guys done for 2024?
We're done for this year, yeah. So that works in an undergraduate lab class that only runs in April and May, like in a 4-week block. So we tend to just make compounds at that time of the year for that reason. It's part of the undergraduate lab. So I decide in around December or January what we're going to make that April in time to order chemicals to the bottom of the world, problems not everyone has. Yeah, so I'll be able to tell you in January if we're making any others. There's actually a few based on the SAR from the paper we're drafting. There's a couple of sort of gaps that I don't think we need to make. It won't hold the paper back, but there's definitely a few compounds there still worth making that will probably be quite potent based on what's working.
Would you want other people to make those if there's only a handful? Like, would that be helpful?
Yeah, if someone else wants to do it, that's fine. It's very simple chemistry. We actually tried to make one of them, it didn't work, but I just think it was due to compound polarity and the students missed it on the column. I think the reaction probably worked, but since it was an undergrad lab, that was their lab report, so that was that. Yeah, I could maybe make a separate post. Depending on timeline, maybe it's good just to make another 3 or 4 and get some data for the paper, but we'd have to ask Wendy. I don't know what their testing situation is and how quickly that could be added. You know, we don't want to hold up the paper they're planning just for a few compounds.
Yeah, I mean,
it's that type of thing is not compulsory, but if you get a real anal reviewer, they might be like, oh, well, that's the obvious one to test, you know.
Yeah, as far as I'm aware, there's nobody doing synthesis at the moment except with large groups of students.
Yeah.
But you never know. We might, have an honors student or something who's interested in doing them sometime earlier than we think.
A summer student in Australia or New Zealand is another thing you could get someone doing.
Yeah, exactly. And if you could put those ones that are like, it would be good to have these 4 or 5, whatever it is, in a separate post. Could you put them on the other molecules to screen repository? Or link,
if you send me the link.
Them? Yeah.
I wouldn't know where to find that.
Yeah, I'll put the link. I'll put the link on the issue that this agenda is on so that everyone can see it. But I think it's fine if you put it either in the Series 5 repository and then we can link to it in the other one or the other way around, just so that, you can find it in either direction, if that makes sense.
Yeah.
Okay. And yeah, I don't think we've heard from Charlotte at ICL in a while, but she would have talked to Wendy directly. And yeah, obviously Wendy isn't here.
I exchange emails with her for a bit because we are both working on a different DNDI project a little bit, like some quite different molecules. So she's still around, like she's still active, if you know what I mean. She's still answering emails and doing chemistry. Yeah.
I mean, I guess if she sent us a batch in February, then there's not that much else to say at the moment.
And she'll definitely need to be involved in the manuscript because although it's the same type of reactions they did, I think they use different coupling reagents and conditions. So in the experimental, there'll be like method A, method B, and so obviously they'll need to write that up.
Yeah, okay. That's a good point. I'm sure Wendy is on top of that, but yeah.
I think so, but I'll post that in the paper thread as well.
Yeah, good idea.
Yeah.
Okay.
I had found the comment from Lori. It was in August.
It was in the August meeting?
No, it wasn't in the August meeting. It was in the Series 3, and it was on August 6th. I had posted a link to it.
Okay, so that was ages ago. That's... I guess maybe we can expect those compounds soon, then.
We can only hope, right?
Pardon?
Let's hope. Fingers crossed.
Yeah. Yeah, okay. Well, I might try and follow up on that and see if she has sent them, because I think normally she would have said that she has. Okay, Series 6. Dimitri, I saw that you've updated the SAR. Is that right?
Yeah.
Diagrams.
I'm gonna just give a short presentation on the results. Is that okay? Fine. Yeah. So I just want to briefly comment on the latest results and on the general SAR trends for the Series 6 scaroxymes that have not been presented yet at the Mycetoma meeting. Just a quick recap for those people who are not familiar with the topic. So I'm working on Series 6 phenarmols, which are a pair of diastereomer isomers, which are synthesized as E and Z isomers. And basically, the caroxyme core looks like that. It is formed from two aromatic rings. The pyridine ring is connected with the CH2 linker, and then we have the active oxide moiety with an variation group, which is termed R in this case. The heat map that we're looking at the moment is refers to the in vitro results, and it essentially shows the potency of those compounds in a in vitro setting before the in vivo testing in larvae models. So the last batch consists of approximately of surplus of 40 compounds. Mainly, most of them were active, with few exceptions, and they can be grouped into several subgroups based on their structure. So essentially, we've been testing straight hydrocarbon chains. Then we went on to cyclic structures, and then this was followed by a series of benzyl compounds. Moieties are groups with different substitutions, but mainly at the fourth position. Coming to the main conclusions, we can definitely say that there is a big difference between the two isomers. So whenever possible, the isomers were separated. Essentially, the compounds were synthesized originally as a mixture of EZ isomers with few exceptions, and they had to be separated, which was not always possible. But apart from that, one of the main conclusions for this series is that we managed to came across is that the E isomer is actually more potent than the Z one. And this can be very perfectly be illustrated by those matched pairs where we have both separated isomers. So for example, dioxylane ring, the E isomer is active, the Z is inactive, and the pattern follows for the other compounds of such kind. So this is quite important because it indicates the confirmation that the E isomer is a better confirmation for the CYP51 enzyme target. The potency values, the percentages here represent the larvae, represent the growth of the hyphae or at 125 micromolar concentrations. So for the E isomers, in most cases, they were, in all of the above mentioned cases, they induced full inhibition of the pathogen, whereas for the Z isomer, in most cases, it was either completely inactive or even gave a surplus growth of the fungus, which is highlighted by the numbers which exceed 100% growth percentage. I'll just come back to the heat map just to illustrate a few other important points. So for example, the same pattern applies to the dioxylane ring. So for example, the Z isomer is inactive and the E is active. Other important findings from this heat map are the fact that we have been extensively studying the ring fluorination, so the benzyl ring fluorination, and it has been found that, for example, the 1-fluorine at the meta position gives an inactive catexime analog, whereas the position at the second, fourth, and 2,4 substitution, including the pentafluoro analog, were all potent, even though not all other isomers were obtained. So there is still some future work to be done if we're talking about like the potential of the series. Also, I wanted to highlight the fact that, for example, we tested oxyconazole nitrate, the Z isomer, which is a commercially marketed antifungal drug, but it's formulated as a nitrate salt. It has been tested against the in vitro as well, against the hyphae, Madurella mycetomatis, and so far it gave the best IC50 and IC90 results. The closest analog that was made is a compound named 104E, which essentially has the same structure to the oxyconazole, but instead of imidazole ring, we have a pyridine ring, which has been extensively studied in both series 1 phenarmals and catechozymes as well. But again, 104 was obtained only as an E isomer, so it's a bit redundant in that sense because there is no direct comparison, and oxyconazole nitrate also is a nitrate salt, so maybe in the future it will be useful to test this as a free base and also obtain the 104 as a Z configuration. But it was synthesized as only as an E isomer and obtained and purified. Also, it's a bit extensive, Clementine, but I'll try to be as quick as possible just to finish off a few things. Also, an important consideration was I generated a graph of log D versus IC50 values for the 30 active catechozyme analogs because we did the same for the phenarmal paper and for series 1 phenarmals, and it was found for the catechozyme series, the one that I just showed in the previous slides. So the trend is not firmly established, but again, at the quadrant in the range of 4.5 to approximately 5.4 log D values at the physiological pH of 7.4 gave the best results for those active compounds so far, which is another important finding because it does not directly correlate with series 1 phenarmal findings, where it was found that ideally log D values should not exceed 2.5, with few exceptions, but mostly 2.5 or less. So series 6 catechozymes were much higher in that sense. Significantly higher, I would say. So this table scatter plot refers to the active compounds that went on to the next stages of testing. For in vivo results, only 4 compounds were tested, even though we had a separated set of E and Z isomers, but only 4 compounds were tested, and those were only formulated as EZ mixtures, unfortunately. Not very high results in terms of larvae survival, to be honest, but the third butyl group, the analog 52EZ, was also quite potent and gave probably the best result. But again, we don't know exactly which of the 2 isomers is giving the potency in that sense. But given the fact that there is a calculated ratio of 9:1 and the E is the prevalent one, and based on the fact that the in vitro results show clearly a trend for the E isomer being the most active, it's very likely that the E isomer is giving the E isomers in those compounds is giving those results. But this would need to be verified in the future if we were to be absolutely sure, because in certain exceptions, there were cases where the Z isomer was active and the E was vice versa inactive in that sense. And that's probably all I wanted to share today. So this kind of concludes the SAR for the series 6 catechozymes.
I just have one question about the larvae survival one. Those are percentages of larvae surviving.
Yeah.
Do you have, numbers, etc., comparing it to the controls or anything, like, significance, that kind of thing? Or is that just like 4 larvae survived?
So it's 12.1% from the overall numbers. So when they do control, so I talked about this with Wendy and Ma some while ago, and they said that, of course, when they inject the control, the survival is 100%. So, but bearing in mind, just a quick recap. So, for example, for series 1 phenarmals, those were the HITS compounds from the DNDi EpiChem library. So phenarmal survival rates were for the most high scoring compounds were in the range of 36.7 to 10%.
Okay,
that's different series, but I'm just giving you the figures
so that's good to know.
that you have a rough understanding in that sense. I think Hunk had a one compound with this scaffold, which is similar to EPL 178, and his survival rates were 39%, and that was so far the highest in vivo result we got. For caroxymes, the 52 EZ is the highest so far. But the problem is there are much more compounds that have been
that haven't made it to the in vivo
purified, separated as diastereomers and not tested. So we still lack a lot of data, and even though I don't think they were planning to test them in the future, it would be I still believe that it would be very useful to come back to this and get some more data. Because in my personal opinion, it feels a bit inconclusive at the moment.
Yeah, I mean, I think we'll have to leave that discussion for when Wendy, etc. are here. I also saw in the chat, VJ, you were saying you could use, you could do some QSAR analysis. What kind of activity data do you need, and how many compounds do you need to do that kind of thing?
Hello, am I audible? Hello.
Hi. Yeah.
Hi.
Yeah. I found that you have IC50 values for 215 molecules, and that's more than sufficient to carry out QSAR analysis. In fact, I can go for explainable artificial intelligence too. It's possible for me, and I can generate atom-to-atom contribution for each and every molecule. This is also feasible. One more thing I found that, one more thing I found in figure A9, there is a graph between IC50 and log D. And, as per the convention of QSAR and this AI, instead of using IC50, we use PIC50. Okay, because we are using log D, that's logarithm. We are normalizing one data, not the other one. So it should not be the graph between IC50. It should be PIC50 versus log D. That you can get better results. This is my opinion. Secondly, I have developed pi descriptor. With the help of pi descriptor, we can calculate more than 58,000 descriptors for every single molecule. So it's a huge data, and we can identify which part of the molecule or which actually chemical properties influencing the final outcome, that is IC50 or MIC or for any other molecule. Okay, this is my humble request. If you provide the data, you can get additional results. Not only SAR, we can go for QSAR. Please, over to you.
Yeah, thank you so much. That would be very useful. And how can we reach you on GitHub, or are there any contacts available?
Yeah, if you can provide the data and structures of the molecule, that much is required to me. I can carry out here.
Okay, thank you so much. I will definitely do this, and
yes, I just need the data. Even if you are able to provide this Smiley's notation for a molecule with IC50 values, rest is mine, and you will get the quality results in very, very soon.
BJ, thank you so much. We'll definitely.
Have you seen the master list? Technically, that has the IC50s and the structures of anything that has an IC50 that we tested.
Please.
Okay.
But we'll generate a separate document, maybe an Excel file with all the caroxymes relevant for the analysis. For quicker navigation. The IC50 values that are depicted in this scatter plot that have been provided by Wendy and Ma. So that's why I included them, just to see the pattern. So of course there is more field for work in that sense. So for further analysis, and the number of molecules is higher than 40. So the things that you see here is just the final batch. So it's much higher than that. So we're talking about 70 novel molecules in the caroxyme series, approximately.
Sorry, I think we should probably just move on, because we've only got a few minutes left.
I'm done with the presentation, so anyway.
Okay, excellent. So unless anyone looks like the main thing left is the kinase inhibitor library. Tim, I don't know if you want to tell us a little bit more about what you're planning to do there.
Yeah. So I haven't really started with the kinase stuff, but I can just quickly tell a little bit more about what I'm going to do as a postdoc here in the group I just had. very brief introduction before. So I'm trained as a molecular microbiologist, working with different fungi before, and now going to work on mycetoma. And so my focus will be on identifying targets that are involved, like associated with the grain formation process. And yeah, so there's already some data from an infection model with the mycetoma, Madurella mycetomatis one, and we will try to also generate a few more with the different species to be able to compare all of the transcriptomic data and look at kinases and some other pathways. So yeah, and then later on, that's also one of the focuses I have, is on extending the genetic toolbox we have for modifying. So if that all works well, we should be able to, generate some knockouts and get experimental proof on if whether or not and how they're involved in grain formation. So I think that's all I can tell for now. I just started last week and all, so yeah.
Yeah, that's perfect, seeing as we don't have that much time anyway. Again, I'll just go through these things quickly, unless someone would really like to expand on them. So Matt and Wendy, obviously not here to tell us about their discussions with this Chapel Hill collaborator, but I haven't heard anything about finding a pharma contact for MycetOS so far. And again, I think these are not really...
Maybe I have a suggestion, because we have so many compounds which are too toxic in the larvae, and so they are just waste or crap compounds. But such compounds which are antifungal and anti-larval might be good compounds as pesticides. So maybe we can also ask chemistry companies which are focused on agrochemicals or pesticides, which might use these crap compounds, I would say, for their projects, and maybe it's possible to generate some funding in this way for testing or for labware funding and so on. Because our group had some cooperation with Bayer Crop Sciences decades ago, and that was quite good to have some money for our own, for chemicals, for labware, for equipment we could spend then, or for PhD students who might be paid in this way then.
That sounds like
maybe we can find some company which might be interested to join, and maybe which has also expertise in the field of pharmaceutical chemistry then. Bigger companies, for example, Dow or something like that, which might be interested to fund the project one way or the other. That was my suggestion now.
That would be wonderful.
Might ask some companies if you want.
Yeah, do you have any, if you want to write down any of those suggestions, yeah, I think certainly we can talk about that next meeting, and then maybe try and get the ball rolling once we've got some other people here to give their suggestions as well on who to contact.
So if they have any, if we do get an agrochem partnership, it's possible that it could work the other way too, because they could have fungicidal compounds that could be used as inspiration for work on our end, right?
Yeah, that would be, it would be wonderful if it worked out that way. We can cross our fingers.
I also found a box that contained not just antifungal agents, but many fungicides that are used on plants. And I also planning to test them to see if there's anything that hits that's not an antifungal agent.
Yeah, and that's in addition to that 6,000 drug library that Wendy was previously screening. which we don't have the results for. Well, Wendy has not shared the results for that yet.
Yeah, sure, that might be also a possibility then if we have a contact in this field that they might provide us with their compound libraries they have as agrochemicals, as fungicides to provide and to vent it for testing then.
Yeah, that would, well, it'd be great. We don't have much time, so I will save this for next meeting. But there's also a group, I forgot to add this to the agenda. There's a group that I am hoping to contact to ask for some compounds as well. So hopefully we'll have lots of things to screen in the next little while, like for new leads, etc. Anyway, it is 9:57 or whatever the corresponding time is in other time zones. So does anyone have any last things to add, or shall we wrap up? Yeah, looks like we're ready to wrap up then. Okay. Well, I will see you all next month, I guess. Our meeting will be at 12 or 1 for Europe and England, and I'm not sure if the time zones have changed again by next month because there's daylight savings, but it'll be a late night in the Southern Hemisphere.
It's going to be 22nd of October next month.
So I think in Australia, the daylight savings will have changed. So it will be an hour different. I don't know what New Zealand does in daylight savings.
Yeah, I think it'll be my 1 or 2 a.m., so I'll probably give it a miss.
Yeah, fair enough.
Yeah, it's a bit too much of a late night.
Yeah, okay. Well, see you guys then.
See ya. Thank you.
Bye. Thank you.
Bye, everybody.